Hi. Hello. This lecture follows the heart anatomy lecture, and this lecture will cover the topics of blood flow through the heart, the cardiac cycle, and the conduction system. The next couple of slides are listing the instructional objectives, and I highly recommend you use those as your study guide. So let's get going on the blood flow. So what goes through the heart is two things. The deoxygenated blood and oxygenated blood both are going to cycle through the heart. And I start the story. I like describing what's going on by starting with the deoxygenated blood and how it arrives on the right side of the heart to the right atrium, and how this blood is meant to go to the through the pulmonary circulation to the lungs. So it goes through the lungs and the pulmonary circulation for reoxygenation. And once it gets reoxygenated, it will go back to the heart, on the left side of the heart, because the heart has the job of pumping that oxygenated blood to the rest of the body. And then it ends in the systemic circulation and is distributed across all the tissues in your body to basically provide the oxygen nutrients necessary for all the cells. So in the PowerPoint version, there are videos throughout, so don't hesitate to look at those. Okay. So we're strictly just talking about the flow of the blood, and I like to start the story at on the right side by talking about the deoxygenated blood. It arrives via the superior vena cava, the inferior vena cava, and the coronary sinus. Coronary sinus also provides deoxygenated blood back to the right atrium. So what is this? This is the blood coming from the head and neck, superior vena cava, and the upper limb, and it's all of that arrives over here into the right atrium, and lower limb, trunk, the rest of the body, all of that deoxygenated blood is arriving via the inferior vena cava, and the deoxygenated blood from the heart itself is arriving via the coronary sinus. So all of this is being dumped into this right atrium. Okay, so we're going to see how this blood ends up being in the right ventricle, but it's going to flow down the right ventricle via these first atrioventricular valves that are called the tricuspid. And so it ends up in the right ventricle, and the right ventricle has the big job of pumping this deoxygenated blood up and out to send it to the lungs, because remember, this is deoxygenated blood, so we need it to be reoxygenated. Where do we do that? In the lungs. So via the pulmonary trunk and across the pulmonary semilunar valve, the blood will get pumped upwards by the right ventricle into the pulmonary trunk and distributed to the lungs, right and left lungs, via these pulmonary arteries. Remember, the terminology for the heart is that all the vessels that are leaving the heart are called arteries, whether they're carrying deoxygenated blood or oxygenated blood. So here it's deoxygenated blood, but we still call it an artery. This is really only at the level of the heart. Everywhere else in the body, arteries are carrying oxygenated blood and veins are carrying deoxygenated blood. I just want to remind you of that. Okay, so the pulmonary trunk splits into pulmonary arteries that will take this deoxygenated blood to the lungs. And when we get to the pulmonary system, I think at the next module, we will talk extensively about what happens at the level of the alveoli and the gas exchange when CO2 will be, delivered to the alveoli so it can be exhaled, and oxygen will be captured there and basically reoxygenate the blood. So the reoxygenated blood will then come back to the heart via these pulmonary veins. All right, so I jumped the gun a bit here. I found this illustration to be quite nice for the pulmonary circulation. Usually it's not really shown when we talk about the blood flow through the heart, and we just say, oh, it goes to the lungs and it comes back. So I just wanted to show you that this is deoxygenated blood going to both sides of course, the two lungs, and there will be capillary beds that are going to basically, we'll see that in the pulmonary system. So I'll save that story for then. But it's in those capillary beds that we're going to see that exchange happening. It's in this direction with the CO2 going out into the other. All right. Yeah. Okay. And when it gets reoxygenated, it comes back via the pulmonary veins. That's what this PV is standing for, pulmonary veins. And again, we use the term veins because these are vessels that are coming into the heart, just like vena cava, superior and inferior vena cava. Those are more veins, or whether it's oxygenated or not. So this is oxygenated blood coming back via these pulmonary veins. Okay, where are we coming back? We're coming back into the left side of the heart. And you can see in the left atrium, you have all of this blood kind of accumulating in the left atrium. And at some point, it's going to drop down into the left ventricle. And we're going to talk soon enough in this lecture later on this passive ventricular filling, which will be followed by an active ventricular filling. Okay, then the left ventricle, at some point, will get the signal that it's time to contract, and the left ventricle will pump this blood up through the aortic semilunar valve up into the aorta. This is the aorta. This is the beginning of the aorta here. And it turns into an aortic arch, and then it goes back down as a descending aorta through the thorax. And we already talked about the branching of the aorta and the cerebral circulation. But that's what the left ventricle is pumping the blood into. It's pumping the blood into the aorta via the aortic semilunar valves. Okay, so you need to know the names of all of the structures, the names of the vessels, the arteries and the veins, the names of the valves, whether they're semilunar valves or the atrioventricular valves. You need to know, the order of that flow. So once it gets pumped out of the aorta, it goes into the systemic circulation. And it's meant to go feed the tissues. So it will arrive at capillary beds in each region that is supposed to go to, which is the entire body. So it arrives at these capillary beds. It's going to slow down. We'll talk about blood velocity a little bit later. It's going to slow down, and it basically gives time for this oxygen to be delivered to the cells and for the blood flow to kind of pick up all the carbon dioxide and metabolic wastes, and then back into the venous circulation and up back to the heart. So we'll talk about vessels and their structures and what makes them different, arteries versus veins. This is a separate chapter that I'm going to present a bit later in a different lecture. All right, and if you're like me, you like the diagrams more than an actual just box of texts. This is the diagram to go with the blood flow through the heart. And I basically kind of verbally described it already. So let's make sure I didn't forget anything. Superior vena cava, inferior vena cava flows through the right valve, right into the right atrium through the AV valve, which is the tricuspid. On the right side is the tricuspid. And then the blood flows into the ventricle, the right ventricle, which pumps it up to the pulmonary trunk. Yes. Yes, I already said all that. So, of course, I use the story of we're coming on the right side and it's deoxygenated blood, and I'm following this particular blood and it's coming back on the left side. But really, this pumping is taking place for both sides. So every time the atria contract, it's both sides that contract. Every time the ventricles contract, it's both sides that contract. So just keep that in mind. So as we're pumping deoxygenated blood to the lungs, we're actually also pumping oxygenated blood to this systemic circulation. Okay, so I included a diagram for you to kind of use to practice. You can use it to, label the different sections of a heart. You can use it to practice the flow of the heart. You can also use it for the conduction system, drawing the conduction system in, maybe a third color, like green, which is actually what I did in an upcoming slide. All right, so that's kind of my way of showing you how you can kind of draw this flow and just make sure you know it. All right, deoxygenated blood on the right and oxygenated blood on the left. Okay, so now that we know the flow of the blood through the heart, let's talk about the cardiac cycle. So the cardiac cycle has to do with we're going to talk about the contraction of these different chambers. What time, when do they contract, and what stimulates their contraction, and the sequence of these contractions. Are they just contracting, randomly, very specifically, in a specific order, actually? So that's what we're looking at now. So what we're going to see is that blood pulls into the atria like this. And whether it's coming from the right or coming from the left, from either veins, it's arriving in the atria, and this blood fills up the atria. And at some point, the pressure in the atria is going to be enough for the valves to swing open, and this blood's going to fall downwards. And that's just from gravity. Gravity actually allows this blood to go downwards. And I'll probably mention it later also anyway, but I can say it now. 80% of the blood ends up in the ventricle without any contraction of the atria. So most of the ventricular filling takes place via passive, a passive process, which is gravity, letting the blood go downwards. Okay. So once this 80% of blood is in the ventricles, there's a remaining 20% that we just need to squeeze out. And that's what happens. The atria are going to go through contraction and squeeze a little bit of blood that's left, because not all of it went through even by gravity. So we just want to make sure that as much blood possible ends up being in the ventricles. And that's where the atrial contraction kicks in, because we want to make sure that everything is out of the atria and into the ventricles. So that is this, let me check this out. That is the additional pressure from the contraction process to squeeze the blood in. Now, so throughout that filling, the AV valves were open. Of course, because we want the blood to get down into the ventricles. The semilunar valves are closed at that point because they don't need to be open. The ventricles are empty. They were empty, and when they're filling, there's no need to have them open yet. So at one point, the ventricles are going to be full, and they're going to start the process of contraction. And just the pressure rising in the ventricles for the upcoming contraction and ejection of the blood upwards, that increases the pressure in the ventricles. It increases the pressure in the ventricles, and that actually helps close these AV valves. It shuts the AV valves. When there is all this blood in the ventricles, they literally, because of the structure of the AV valves, it kind of fill the cups of these AV valves and kind of shut them closed. Plus, we also have the papillary muscles that are pulling on these chorda tendineae strings, these very thin connective tissue strings, and it pulls on these strings and it shuts these semilunar valves. These AV valves. So these AV valves, they kind of come together towards the center and they get shut like that. So they get shut, which is great because we do not want backflow into the atrium. That's the main purpose of having these valves, is to prevent the backflow into the previous chambers. So now we have ventricles that are full, and the pressure is going to be rising in here. The semilunar valves are still closed. The AV valves just closed. And at this point, we call this an isovolumetric contraction, where all the valves are closed, but there is this contraction that is raising the pressure in the ventricles. Okay, when the pressure is high enough that it's even higher than the pressure in the aorta, finally, the blood can push these semilunar valves open, and you can see that here. These semilunar valves are swinging open. So the semilunar valves are like saucer cups. They're the opposite direction of the AV valves. You see these are like upside down cups, but the semilunar valves are right side up. And when the pressure in the ventricles is higher than the pressure in the aorta, we can finally push that blood through and get it into these big arteries, whether it's the pulmonary trunk or the aorta. And I wanted to make sure you realized there's blood in here, even though the diagrams, rarely show the blood in the aorta or the blood in the pulmonary trunk. There's blood in there. It's not empty. There's blood in there. So there's pressure in these arteries. We just need the ventricular pressure to be bigger than the arterial pressures above, so we can actually get through the semilunar valves and push that blood through. Okay, so at one point we will have this pressure higher than the pressure in the arteries. Mainly we talk about the aorta. So we can actually push this blood through the semilunar valves and into those arteries. So at that point, the semilunar valves are open. Of course, we're trying to, get the blood to the next, area. And here it's not a chamber per se, but it's the arteries. And so once that blood got squirted up there, it actually falls back down when the pressure of the ventricle drops, right? The ventricle just got empty. All the blood got squeezed upwards. And the ventricles, because they're empty now, they like they deflate, like they relax, and so there's this pressure drop, and because of the pressure drop, we have this backwards flow of the blood, and this backwards flow of the blood actually helps close the semilunar valves. I mentioned that in the heart anatomy lecture when I talked about the semilunar valve structure and function. And these semilunar valves have these cups. And when the blood gets in the cups, it actually brings these cups close together and shut them closed. So it's really cool how this works. Like this. And I don't remember if I mentioned it in the when I talked about the coronary circulation, the carotid, the coronary arteries that are coming out at the base of the aorta, these coronary arteries, they when the blood goes to the coronary arteries, it's actually during that drop in pressure in the ventricles, because the blood comes backwards. Of course, it helps shut the semilunar valves, but it also goes into the coronary circulation. So that's when the blood goes into the coronary circulation, when it falls backwards and it just kind of like gets into the coronary circulation, right? Because that's the beginning here of these coronary arteries, right and left coronary arteries. So that's when the blood is injected in the coronary arteries, when there's that backflow. Okay, at this point, the semilunar valves are closed, and really the AV valves, they should be shown closed. Yeah, they really should be shown closed at this point here, especially that there's no blood in this diagram. So at one point here, the semilunar valves are closed, and the AV valves are still closed too. So, but the ventricles are relaxed. So we call this an isovolumetric relaxation, where there's no change in volume of the blood in these chambers, because we're basically waiting for the next step. Okay. So I mentioned the papillary muscles helping kind of shut these valves close, the AV valves, when there's this negative pressure in the ventricles. Okay. So as I was describing all this, I didn't introduce the terms systole and diastole yet. So those terms refer to the process of contraction or relaxation of that cardiac muscle. So when the heart is pumping, when it pumps, we call this a systole, because there's contraction of the muscle. And when it relaxes, we call this a diastole. And that's when the muscle is relaxed. So the chambers, we say it's a ventricular systole when the ventricles are squeezing and contracting to eject the blood upwards, or we talk about atrial systole when the atria are squeezing, and when they're not squeezing, we talk about diastole. Keep that in mind, that blood moves through the circulatory system from areas of high pressure to low pressure. And it kind of makes sense, because here you are trying to squeeze the blood through a vessel. So you're increasing the pressure at one end to kind of like push it forward. So there's less pressure at the other end, and you're trying to push that blood forward so that the higher pressure is at the end of where you're starting, and you're moving that blood forward like that. So the blood moves from areas of high pressure to low pressure. And that's what this contraction does. It creates pressure. Imagine you're contracting this, like, let's say this is a balloon, and it's completely closed, you're squeezing on the surface of the balloon, and it's really like stretching the edge of the balloon. So at one point the balloon might pop. So here we have valves to open when the pressure is high enough that the blood can allow the blood to flow forward to the next region. Okay, so there are 4 phases to the cycle. There is the ventricular filling. That's the first phase. So that's when the story starts, where we have the atria that are dumping the blood down into the ventricles, and that's called ventricular filling. And I already mentioned it starts actually with a passive filling, and it's quite fast. So that's called a rapid filling, and it's a passive. This is passive. It's by gravity. And then we have active filling of the ventricles. That's what we're talking about. When the atria finally decide, okay, we were able to get as much blood downwards, and now it's time to squeeze just to make sure everything is down there. So there is what we call atrial systole, squeezing of the atria to allow as much blood possible into the ventricles. So then the next phase is called, so this is number 2 here, this is called the isovolumetric contraction. When the ventricles are starting to contract, so they're going to get into systole, they're starting to be in systole, but the valves are all closed, so there is really no movement and no change in volume. It's just in one chamber, the blood is there, not moving, but there's pressure rising because those ventricles are squeezing. So that is called isovolumetric contraction. Finally, when the pressure is high enough, as I mentioned earlier, we have ventricular ejection of the blood into the arteries. That's during ventricular systole. So this is ventricular systole. Just like we wrote atrial systole here. Ventricular ejection takes place during ventricular systole. And then finally, the ventricles relax, right? They're relaxed here. And that's ventricular diastole. And the atria are already relaxed because they weren't squeezing anyway, even before that. So they're already relaxed. So ventricles are relaxed, everybody's relaxed. So this is isovolumetric relaxation. There is still no change or movement of blood from any chamber to any vessel or from chamber to chamber because the AV valves are still closed. The AV valves are still closed, the atria are filling, the ventricles are relaxed, atria are relaxed. There's no movement of blood. This is called isovolumetric relaxation, and that's, that 4th phase. Okay. I really don't need to redraw it here again. So I'm just going to skip that. You can use it for your own purpose when you're studying. So let's look at each one of these phases. That's what I'm doing here, is I'm doing these different phases one at a time. I'm looking at one of them at a time, right? I'm going to look at number 1 first, then number 2, number 3, number 4, the phases of the cardiac cycle. And I might be repeating myself on certain things because I kind of already mentioned all of the words. And I think this is just then the final summary here. So passive ventricular filling, that's the first thing that happens when the blood that pulled into the atria, coming from, superior, inferior vena cava, the coronary sinus, and the blood fills. At one point, it's going to be heavy enough, you can think about it like that, heavy enough that it's going to open these valves. Imagine it's like a door at your feet that if you're heavy enough, you can actually kind of fall through that door. So these valves are going to open. The atrial pressure rises above the ventricular pressure. So there's more pressure above here than in the ventricles. So the blood flows into the relaxed ventricles, accounting for, we usually see 80. I just read in the textbook and other sources, it's usually 80. So AV valves open, semilunar valves are still closed because they have no business being open right now. Okay. So the active ventricular filling is so the second phase. So that's the second phase of that ventricular filling. We had the passive ventricular filling. This is the active ventricular filling. This is when we have systole. I don't know why there's the word diastole here. This is bothering me. Let's go back to here. Yeah, it should have been systole. This is in the active portion of the ventricular filling. There's a passive portion and an active portion, and the active portion is your final squeezing. So this is an unfortunate typo, but that's okay. I caught it, and that's what matters. Active ventricular filling is when the atria are contracting. So we'll talk about the SA node, the pacemaker cells that are sending the signals to the atria, telling them, come on, guys, we have to make sure that we got as much blood downwards. So this is the P wave on the EKG. We're going to see that, and that completes the ventricular filling. So technically speaking, if the atria weren't contracting, the passive filling, because it takes care of 80% of the blood, the passive filling is still okay. So contraction of the atria is not necessary for heart function, technically speaking. But during exercise, it would be very important to have atrial systole to make sure that there's as much blood possible at every beat, because now the blood is really, really needed throughout the body when you're exercising. Okay, so then we have the systole, ventricular systole, that is going to start, and this is pushing the AV valves to close, and the semilunar valves are still closed. And that's why we have this isovolume. The volume does not change. There's no movement of blood from one chamber to the other, but we are already contracting here. We are contracting the ventricles. So we talk about isovolumic contraction. Actually, in the textbooks, it says isovolumic or isometric or isovolumetric. I saw the 3 ways of saying the same thing. So just to let you know. So when these ventricles contract, they are really increasing the pressure, and eventually it will be bigger than the arteries above. But this pressure increasing is what closes the AV valves. And so that happens like right away when the ventricle starts going into systole. And the semilunar valves are still closed at that point because the pressure is not high enough, but it's going to get higher and higher. Okay. Remember that number. It's 120 milliliters usually, the amount of blood in the ventricles that came from the atria. This is called the end diastolic volume, and we're definitely going to see that again when we talk about cardiac output and stroke volume and all of that. This is another lecture that will explain a lot of that. But here's an introduction to the end diastolic volume. It's basically the volume in the ventricles, volume of blood that's in the ventricles that was basically given by the atria. And that's it. We're not changing that volume anymore. All the doors are closed. There's no changing your mind. This is it. You're here in the ventricles and you have, in general, 120 milliliters of blood in that ventricle ready to be ejected through the arteries. So that's what we call the end diastolic volume. It's the volume of blood at the end of diastole before the ventricles start contracting. So when the atria relax and they're done contracting and all the blood went down into the ventricles, that's the end diastolic volume. That's basically the volume of blood you have in the ventricles that hopefully you'll eject as much as possible out. And we'll talk about ejection fraction and all of that. All right. So finally, when this pressure, look at this, this is really squeezing. So the pressure is very high and it's going to push these semilunar valves to open. Pressure in the pulmonary trunk and aorta. So yes, so the pressure is going to be higher than in the pulmonary trunk and aorta. This pushes the cusps to open. Blood is ejected. The pressure in the two ventricles are different. Yes, the pressure in the left ventricle is around 120 millimeter mercury. In the right ventricle is much, much lower. We don't have to go as far. We just go to the lungs. But 120 millimeter mercury is so we can shoot that blood as far as possible as your pinky toe, where it also needs to be supplied, right? All the skin cells and muscles in your pinky toes. So that's. So after that first initial, this is like the highest. This is your systolic blood pressure. This is the highest blood pressure you can have. It's right there at the aorta, right when the blood is ejected. And as soon as it's ejected and it goes out, that pressure is going to drop back. So at the end of the period of ejection, there is around 50 ml of blood left behind, and that's called the end systolic volume. The end systolic volume is what's left in the ventricles. So basically, what you ejected is your ejection volume, your 50 ml, and what you're left at the end of the systole, the ventricular systole, what's left at the end in the ventricles, because it's not 100%, it's the end systolic volume. Sorry, the 50 ml is what's remaining, and what's ejected, I believe it's around 70 ml. So you try to pump as much out. I think it's around 60%. We look for that ejection fraction, it's called, where 60% of it is going to be ejected and 40% will be left behind, because that's all we can do. So then when that's done, everybody relaxes. That's the period of isovolumic or isovolumetric relaxation, where all the valves are closed, but we're finally going into diastole for the ventricles. The atria were already in diastole. So that's why this is the time where there's no volume changes. There's no movement of blood at this point. It's just that we're going into diastole and all the valves are closed. So I did forget to mention that every time a valve's closed, I talked about right here. I talked about the AV valves kind of shutting closed from that the beginning systole of ventricles. It kind of like shuts these valves closed. And then I talked about how when the blood falls back down, where was this? Okay, it's not illustrated here, but I showed that to you a bit earlier. When the blood falls back down, right here. Yes, this was here. You know, how it shuts these semilunar valves closed, right? So every time the valves close, they make a sound. So I forgot to mention that. They make a sound. Let's go back to the end here. And that's the sound you can hear. So they're called S1 and S2. And these are the lub dub, lub dub, lub dub. These are the sounds that we can hear in the stethoscope. So I'm going to save the discussion of the Wigger's diagram for a group discussion over a live session. And this will be kind of like a mini assignment to do before we meet live, where I'm going to ask you to understand every aspect of this Wigger's diagram. So we will talk about that on Friday. All right, so the first hard sound is your AV valves closing. That's the first one. And actually, it is on the Wigger's diagram. It shows you here the first hard sound and the second hard sound. And the first hard sound and the second hard sound. This is a little bit of turbulence that's in the aorta that you can hear as a third hard sound, but we're going to stick to first and second. So the AV valves closing makes that first sound, and then the aortic and pulmonary semilunar valves closing make up the second sound. Now the third sound, occasionally you can catch that, is caused by the turbulent blood flow into hypertrophied ventricles. So when the blood flows into enlarged, hypertrophy is the term we use in anatomy when something is bigger or got bigger. Muscle hypertrophy is when the muscle kind of enlarges. Chondrocyte hypertrophy in the metaphysis, if you remember that part of the endochondral ossification, those lacunae hypertrophy, they enlarge. So that's what hypertrophy means. So this is referring to ventricular hypertrophy. So if the ventricles are enlarged, you can actually hear the blood flow through the ventricles a bit differently, because it's not as smooth as if the ventricles weren't enlarged. And you can hear that third sound. Okay. So the next part of this lecture is about the cardiac muscle, the autorhythmicity of that cardiac muscle, the role of the pacemaker cells that really are setting the pace of this autorhythmicity. They're the ones that are autorhythmic and kind of sending those signals. So this is basically this next the next portion of this big lecture. All right, so let's tackle pacemaker cells, cardiac cells, and their and the conduction system. So cardiac cells, if you hopefully remember from histology when we first talked about cardiac cells and skeletal muscle cells and smooth muscle cells, this was a whole tissue category, right? We have 4 tissue types: epithelial tissues, connective tissues, muscle tissues, and nervous tissue. So in the muscle tissues, we saw 3 types of muscle tissues. Cardiac muscle happens to be one of them. cardiac muscle cells are striated. You can see that very, very nicely here with striations. Just like skeletal muscle cells were. Smooth muscle cells were not striated. Striations really only reflect the fact that the contractile proteins are very, very organized in these two types of cells, skeletal and cardiac. That doesn't mean there are no contractile proteins in smooth muscle cells because they contract, don't they? Except that the contractile proteins in smooth muscle cells are not as organized, so they don't end up in a specific kind of like beautiful pattern that you can catch with your eyes under the microscope. So you don't see striations, but the contractile cells are there because in smooth muscle cells, because they have to contract. Anyway, the nucleus is usually one per cell and it's central. Unlike skeletal muscle cells, they are multinucleate cells and they're very, very long cells. Cardiac cells are short, sometimes branched, like this. Right, so it depends, you know. So they're square, they're rectangle, they're branched, and they have a central nucleus with striations. What's very special about cardiac cells, which we also call myocytes, by the way, they have intercalated discs. Look at these beautiful intercalated discs right here, right here, right here, all of these. What is this? This is cellular junction. So the junction between, here, you see that? This, and I didn't draw very standard. That's okay. The junctions between the cells are called intercalated discs. They contain two types of junctions. They contain desmosomes and intercalated and gap junctions. Those are the two types of junctions, cellular junctions that intercalated discs have. Okay. So now let's talk about the conducting system. And then I talk about pacemaker cells first, and then I will circle back into the cardiac cell and discuss the action potential of both of these types of cells. So the conducting system. So you have to know that the heart spontaneously depolarizes by itself. And this is really what we describe as automaticity, or that it's autorhythmic, because it depolarizes. Poof, it depolarizes. So that's, and then it repolarizes. It depolarizes, repolarizes, depolarizes, repolarizes throughout your entire life. And the cells that are part of the heart that are at the source of this depolarization are of two kinds. They are nodal cells. So maybe I will write this on the side here. Actually, I'm going to add a page. I'm going to add a page. And there we go. So I want you to know about the nodal cells. So we first have, so the myocardium is made of two types of cells. The nodal cells are literally 1%. 1% of the myocardium are nodal cells. The pacemakers. Pacemaker cells. Okay. These cells do not contract. Very important. The 99% of the remaining cells are your myocytes, the cardiac cells, and they are going to do a contraction. So the nodal cells, we're going to see there is an SA node, AV node, AV bundle, AV bundle or bundle of his, it's called. AV bundle, bundle of his, bundled branches, right and left branches, and Purkinje branches. And Purkinje fibers. Okay, so I'm going to draw them on the diagram in a minute anyway. So they're the one that sets a rhythmic pace. That's what I want you to not forget. They don't contract and they set a rhythmic pace. So this is the first type of cell. The second type of cell, 99% of myocardium, are the myocytes or cardiac cells. Okay, so these cells, they're contractile. So they are contractile, which means that they have actin and myosin. These are the contractile proteins, but also the regulatory proteins like troponin, tropomyosin. This is very important. So they're contractile because they have the machinery to do that. And that's it. That's all I want to say about that. So these two cells. So now let's look at the sinus rhythm. Here we go. I'm going to use green for doing this conduction system. So we start with the SA node. It's on some books they say lateral, some books they say medial. I believe it's on the lateral side of the entrance of the superior vena cava. It's not a big deal. And these cells, they are your pacemaker cells, and they're all throughout here. So I'm going to draw them as I'm not going to draw them as little circles. That's okay. They also send branches. So this is behind. Let's do this. We call this the Bachmann's bundle. So these branches are meant to go stimulate the cells of the left atrium because the SA node is on the right side. So it's very convenient here for all of these cells that are in the right atrium. But to go stimulate the cells of the left atrium, we need kind of like these more long fibers to go to the other side and stimulate the cells on the other side. And we call these the Bachmann's bundle. And then we have the internodal pathway, which is all of this. This is the internodal pathway. It's a collection of branches that are going to not only stimulate all of the myocytes in the atrium, but the goal is to stimulate the node. That's what's over here at the base of the right atrium. Atrial ventricular. Okay. So as the signal propagates from the SA node into the right and then the left atrium, as the signal propagates, it creates the depolarization of the cells, and the cells are going to contract. This atrial contraction, or atrial systole, as this propagates around atrium. The atrial chambers that are going to contract. Okay. Right. So we arrive at the AV node. There's a delay. There's a delay before the signal goes down the AV node and into this AV bundle or bundle of phase. So it's like a bundle of phase. And this is going to split into bundle branches, right and left bundle branches. And the signal is going to travel to the apex of the heart and actually start moving upwards here via the anyway, so we have the signal going from the apex and working upwards. So when the signal is down there, it's to depolarize the cells down there, so the myocytes of the ventricles. And it actually starts from the bottom working its way up. It makes sense, right? Because we're kind of squeezing this chamber. And if we squeeze from the bottom going upwards, then we kind of like don't miss anything, right? We want all the blood to come out. We don't want to start squeezing from the top. That might push the blood back down. We need to squeeze from the bottom up. So we actually have the signal that's going to propagate from the apex upwards here to the Purkinje fibers. And there were a couple of anatomy diagrams. It's in the textbook. Textbook. Textbook for the other person. Showing you that the muscle cells, the myocytes or cardiac cells, they're actually laying in different directions in a couple different layers, and each layer is different direction. So when they squeeze, actually create this motion of literally bringing the ventricles. Literally, it just is the way the fibers are oriented. It allows for the ventricles to squeeze, and they kind of like they go to each other. So it's really cool. So the idea is to squeeze the blood upwards. Okay. So once that is happening, that's when the ventricular system takes place. Okay. So why are we having a delay here? This is very important. The delay here, let's move it. There's a delay here. And this delay is going to be really important because remember, there is contraction going on in the atrium. The atrial system is already going on. And there's atria that are contracting because the signal propagated across the atria and chambers that are contracting. So atrial system is going on. But we don't want to start the ventricular system quite yet because we want to make sure that the atria really push as much of the blood as possible downwards. So we want to make sure the ventricles fill to the maximum capacity before they start contracting. If they start contracting too soon, if the contraction starts too soon, then it's not going to be as much blood that's pumped out because we didn't give it time to fill. But when there's this delay, we give the ventricles time to fill and to the maximum capacity possible before they contract and inject the blood out by the organs. So the delay is very important. So how is the delay physically possible? Cool. First, there is quite a bit of connective tissue right there. This is at the level of the septum over here. There's a lot of connective tissue for signal. So it's kind of a bit harder to get through. The signal doesn't get through as fast as it was. And second, the diameter of these fibers over here is smaller. So if you remember from graduates about axon fibers, for example, the larger the axon fiber, the faster the signal. The smaller the diameter, that fiber, slower the signal. So here, the fibers actually reduce in size, so the diameter is smaller, and it goes to the delay because it slows the signal down. Also, there are less gap junctions. So the propagation of the signal from cell to cell to cell to cell is not going to happen as fast. And that helps creating this delay, which we need to make sure that it's good. So we can yeah, so cells have a smaller diameter, which reduces the velocity, slower opening of the calcium channels. Oh, I forgot to say that. And less gap junctions. So the calcium channels happen to also not open as fast right there that way. Okay. So one thing I wanted to talk about, because that's really interesting, it's the fact that, so I'm going to put some numbers here, and I have a hard time memorizing all these numbers, so I'm going to look at my notes here. The SA node sets a pace of 60 to 100 depolarization per minute. So I'm going to write here 60 to 100 depolarization per minute. The signal, right? Signal sending signal, 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 100 times per minute. That's 60 to 100 times per minute. The other parts of the atria are 60 to 80. So there's cells that are kind of like distributed throughout the atria that are part of these internodal pathway. We're talking about the nodes, the nodal cells, right? This is all I'm talking about right now. The nodal cells sending the signals. In the internodal pathway and across the atria, they go from 60 to 80 depolarization per minute. The AV node depolarizes 40 to 60 times. So in the AV node, I'm going to write here 40 to 60 times per minute. So that's right? So it's sending a signal 60 to 100 times depolarization per minute, or 60 to 80, or 40 to 60. You see how it's less and less and less. And then the Purkinje fibers right there are actually sending signals from 20 to 40 depolarization per minute. So why am I saying that? And I won't ask you about memorizing these numbers on an exam per se. I just wanted to show you something. All of these other cells, right, besides the SA node, the cells, the still nodal cells that are part of the internodal pathway, the nodal cells that are in the AV node, right, the AV node, the nodal cells that are part of the Purkinje pathway, right, all of these are nodal cells, and they're all sending signals. So technically speaking, we could go without the SA node. And I think I had, this is, I alluded to that earlier. We can go without the SA node. There's actually a plan B for the heart. In case the SA node is not functional, there's actually a plan B. And the plan B is this. These other nodal cells that are in the atria are actually sending depolarization signals that can maintain a pace. So then that would be a new pace that is set by these cells, the internodal cells, or the cells that are part of the HO1, the nodal cells part of the HO1. So there is a plan B, and that's it. That's plan B, in case the SA node doesn't work. And we actually have a plan C. So this is actually plan C, the AP node, and plan A, B, C, D, a plan D. So really, if we needed to, we could count on the Purkinje fibers to be these cells that will set the pace at 40 to 60, 40, 20 to 40 depolarization per minute. Of course, it's not enough. Of course, you know, it's not ideal, and it's still a problem, but the heart is still beating. That's the point. Okay. All right. So let's talk about now some more physiology on these. So yeah, this is the summary of the conducting system, who I definitely already talked about. Okay. So let's talk about really the nitty-gritty of the physiology of this. It's important because we're going to talk about calcium, and calcium is so important for the contraction of the heart. You're going to learn about, you know, if there's too much calcium, what happens to the heart, not enough calcium, what happens to the heart. All of this is pertinent for, you know, for in a medical setting. So this first slide is showing you, basically it's a drawing I made to show you the actors here, and I have on the left a nodal cell, and on the right I have a cardiac cell, contractile cardiac myocytes, cardiac cell, or however you want to call it. And we're going to talk about a several number of ions going in, going out, da 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 da. So I labeled all this here. I might not have all these labels, you know, in a future diagram, but at least I labeled them here. So we're going to see leaky sodium ion channels. These are the first channels we're going to talk about. Leaky sodium ion channels. Right here, leaky sodium ion channels. I'm going to talk about calcium channels. Some are, and they're all voltage gated channels. So when something happens with the membrane potential, where we're tweaking the membrane potential, then we have channels that are so sensitive to the change of the membrane that they get excited. So these channels open up if you change the voltage at the membrane. So these are actually, both of these are voltage gated channels. And we're going to see two types, the fast voltage gated channels and the slow voltage gated channels. I included the fast voltage gated channels, even though probably to make it easier, I shouldn't even mention them, but I think you can handle that. Okay. Of course, we're going to talk about gap junctions and how these ions are going to actually move from one cell to the other. And once they get into the other cell, the cardiac cell, I'm going to talk about the voltage gated channels in that cell. And we have voltage gated sodium channels, voltage gated calcium channels, voltage gated potassium channels that are all going to have a specific job to do when we get to them. So the nodal cells, we're going to start the story with leaky sodium channels, and then what it does to these voltage gated calcium channels, and how potassium channels will open up to repolarize the cell once the depolarization is complete. Okay, so let's do that. So these funny leaky sodium channels, they allow for the leakage of sodium ions. So you have sodium ions that are going to be kind of trickling in. Because as you know from the sodium potassium pump, which I did not draw it here, sodium ions are in higher concentration outside of the cell than they are inside of the cell. More sodium ions outside of the cell, always. And the opposite is the case for potassium ions. The sodium potassium pumps, this is something that should already be anchored in your memory forever. The sodium potassium pump establishes gradients for both of these ions, where you have more sodium ions outside than inside the cell, and more potassium ions inside the cell than outside. And the pump keeps these ions unequally distributed, and that's what contributes to the membrane potential. So if I wanted to draw the membrane potential over here, it would be something like this. This is the resting membrane potential, which is right around -60 millivolts in a nodal cell, right? So I'm going to draw the action potential right here, how it changes as these ions are moving. Okay, so we have these leaky channels, and they're literally called funny. They're called funny leaky sodium channels. I'm not too sure why. They're coming in. So when you're bringing positive charges inside the cell, it makes the inside of the cell less negative, right? The resting membrane potential with this unequal distribution of charges is such that you have more positive charges outside than you have positive charges inside. That's the resting membrane potential. And I have a video in my YouTube channel about me explaining the resting membrane potential, if that is something you still haven't kind of mastered yet. So the resting membrane potential at the membrane of a cell, positive charges on the outside, negative charges on the inside. We actually just say less positive on the inside. So you bring positive charges inside the cell, now this is going to be less negative. It's going to be less negative. So we go from -60 to, so let's keep it with the color coded. So we have a potential that's going to go to be less negative, and we call this a depolarization. Depolarization. Right here, this is depolarization starting. And it's going up, so it's going to be -60. It was -60, it's going to be maybe -58, -57, -56. And when it's at -55, boom, 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 we have the first voltage gated channels that are going to open for calcium. And here we go. And these are fast, and you have basically an injection of calcium ions. Boom. Here we go. So they further depolarize the membrane, and now we go from 55 to -40, -55 to -40. Here we go. That's the threshold. That's the threshold for the actual most important calcium channels. Are these a bit slower, but voltage sensitive because we're changing the voltage at the membrane. At -40, we have these channels that open up and bring so many calcium channels that we have this huge depolarization of the membrane. So many plus charges, right? And it actually goes all the way up to plus 10 millivolts. Wow. So we completely reverse the polarity. So all of these plus charges, I literally reverse the polarity at the membrane. So we would be looking at something like this. The membrane would be depolarized completely, right? We passed 0 and now we're in the plus signs. That means we completely reverse the polarity at the membrane. So these ions are going to be so important to actually stimulate the cell next door. And that's where they're going to end up, because they're going to take the tunnel of the gap junctions and they're going to end up in the neighboring cell, which is your cardiac cell. And that's what we want. We want the nodal cells to stimulate the cardiac cells. They're setting the pace, and the cardiac cells are going to continue that they're going to get excited and they're going to excite their neighboring cells. So the signal is going to propagate via the cardiac cells as well. Remember, intercalated disc is both desmosomes, which are your step buttons between cells, and then the actual tunnel allowing the gap junctions, allowing the passage of ions from one cell to the other. Okay, so eventually this cell, this nodal cell, is going to repolarize because of the exit of potassium ions. So at one point, when we reach +10, we're going to have repolarization of the cell. So over here, we have potassium channels open. Potassium channels open. And right there, oops. So at the peak there, calcium channels have closed. Right here. I don't know how I'm going to do this now. I don't know how to edit this. All right. So the cell depolarized, and then the cell repolarized with the efflux of potassium ions out of these channels. Okay. Okay, so that's that. So then let's talk about what happens next. So I'm going to talk about the depolarization of the cardiac cell using this next graph, this next action potential graph. And so what happens? So we have all of these calcium ions that are coming through those gap junctions. They're arriving here, and what's going to happen is that we're going to see the arrival of these ions is basically helping depolarize the membrane of the cardiac cell. And the resting membrane potential of the cardiac cell is actually even more negative than a nodal cell. We start at negative 90 millivolts. Before we talk about the cardiac cell, I just wanted to say, you see here, for the nodal cell, the action potential, we depolarized, repolarized, and actually it's going to go straight into a second depolarization right away. There is no time to wait. So I don't have room here. Maybe I'll just take it out. And I'm just going to draw in pink. See, and it goes up and it goes back down, and it really repeats itself like that. There's no resting membrane potential that actually stays resting. It's constantly depolarizing, repolarizing, depolarizing, repolarizing. I wanted to make sure I showed that to you before I moved on to the cardiac cells. Okay, so these ions moving into the cardiac cells, they're creating a depolarization in the cardiac cell, and that's that first depolarization I drew over here in pink. So these are your calcium coming in. Calcium coming in. So when the calciums come in, it's going to trigger the voltage sensitive channels, we would say voltage sensitive or voltage gated channels for sodium ions to open up. They open up as soon as they see the calcium coming in at the negative, around negative 70 millivolts. That's the threshold. So I didn't have a chance here to write the word threshold. And this is what this is. This is the threshold. T for threshold. So when there are enough calcium ions that came into the cardiac cell, and we depolarize the membrane from negative 90 to negative 85, negative 80, negative 75, negative 70, that's it. We reach negative 70, these voltage channels open up. And now we have a huge depolarization. Because of the influx of sodium ions. There we go. This goes around plus 20 millivolts. Okay. At that point, what we have so at that point, I think we were way over here. I'm sorry about that. Okay, I'll stick to the one I was drawing. That's okay. What happened? Oh, that's so odd. I have no idea what happened here. It erased everything I did. I just did. Just by moving the slide up and down. That's so weird. Okay, I'm not sure why it erased everything. Okay, I'll continue the story here. It's okay. So as you get in, as the sodium ion get in the cell, we were here at depolarization of the membrane, and we reached plus 20. So just follow me here. Try not to like look at the rest. Let's follow this here. And I'll provide you with all of these drawings at different stages, so you can actually, and one that's completely empty, so you can actually draw it out and kind of like, repeat it for yourself verbally, orally. You can kind of like talk the story to yourself. So this phase is called phase 0, when we're depolarizing the cell and we reach 20 millivolts. We reach 20 millivolts, and at that point, there are potassium channels that open up, and we have a short repolarization of the cell, and this is what's showing over here. Right here. This is a repolarization of the cell because we have these fast-acting potassium channels that open up, and we call this phase 1. It's a short repolarization of the cell, but then the membrane permeability for potassium ions decreases, so it doesn't last long. what picks up afterwards is the opening of calcium channels. And these calcium channels are allowing calcium ions to come in. And because calcium ions are coming in and potassium ions are still kind of going out through these fast channels, even though the membrane permeability is decreasing, they're still going out. So with this potassium going out, calcium coming in, it's like they're fighting for, okay, who's going to depolarize? No, I want to repolarize. No, I want to depolarize. I want to repolarize. They're both moving in and out of the cell. So we end up with a plateau, where there is neither depolarization nor repolarization. So that's why I drew here this line in two colors, the blue to represent still potassium ions coming out, although permeability is decreasing for potassium ions, and calcium ions coming in at kind of at the same rate, and it's create this plateau. That influx of calcium ions, which we call this during the plateau phase, during phase 2, we call this phase 2, that's when we have contraction that is initiated. Finally, we're contracting. Okay. So the calcium that is coming from the outside is very important, but it's not enough. It's not all the calcium that's going to actually be available for the cells to contract. The calcium in the SR is going to come out as well. Remember, calcium is stored in the sarcoplasmic reticulum of muscle cells. Skeletal muscle cells had a very extensive sarcoplasmic reticulum with a lot of calcium stored in there. So that calcium for the SR and skeletal muscle cells, action potential had to come down the T-tubule and trigger the opening of the channels for calcium to come out. In cardiac cells, it's a little bit different. Cardiac cells, the calcium is going to be induced to come out of the SR via the binding of the calcium that's arriving in the cell. So we have calcium coming in, either through the nodal cell or from the outside. That calcium is going to bind onto it's like a ligand-gated channel, basically. The ryanodine is the name of this channel. It has a receptor portion for the ligand to bind on it, and then a channel portion that's going to open up only if there's binding of the ligand. That's why we call them ligand-gated channels or ligand-sensitive channels. When the ligand binds, the gate opens, and the gate is for calcium, and the ligand is calcium. So we say calcium-induced calcium release. So now we're releasing the calcium that was in the SR, and we're letting it out. So now there's even more calcium in the cell. Lots of calcium, good contraction. That's what's going to happen. So I put down here a reminder of where calcium is going to be used. If you remember your what's going on in a skeletal muscle cell, when calcium is released from the SR, it goes and binds troponin. Troponin is a calcium binding protein. Troponin binds calcium, or calcium binds on troponin, and troponin tropomyosin is a complex that is literally hiding actin active sites for binding of myosin. Troponin and tropomyosin are literally covering actin, and actin and myosin are not able to bind together because of this coverage of troponin and tropomyosin. And if you want to review that, I also have a video about that phase of muscle contraction as a video of me explaining that on my YouTube channel. So troponin finally sees calcium, troponin binds calcium, and the whole troponin tropomyosin complex swivels out of the way. I describe my troponin tropomyosin as a door. There's this door that's blocking the way of the myosin heads to bind on the actin filaments. So the door is in the way, but we have a doorknob. So the door is actually tropomyosin. The doorknob is troponin. And you add the key to the doorknob, that's calcium. Calcium gets into the key as the key into the doorknob. And as soon as calcium binds to troponin, the whole door swivels out of the way and basically reveals the binding sites for the heads of the myosin into the actin filaments. And then you can start the cross bridges take place between the heads of the myosin and actin, and you have the sliding filament theory. So I'm not drawing all of this. I just wanted to remind you that calcium goes and binds troponin, and then you have that whole story of the troponin tropomyosin swiveling out of the way for the cross bridges to occur. Okay. So that's what happens when we have calcium in the cell and the cell contracts, and it's great. But now we need, so that was phase 2, right? The plateau was phase 2. In phase 3, we repolarize the cell. We repolarize the cell by allowing potassium channels to open. These are a little bit different from the previous potassium channels of phase 1. These are a little bit slower channels. They allow for the efflux of potassium ions, which that's how we end up with adding these positive charges outside and basically repolarizing the cell. So we're going back to bringing membrane polarity back to, being more negative on the outside and more positive on the outside, more negative on the inside. So we're repolarizing the cell. So now it's going back down. And the goal is to get back to resting membrane potential. What happens to the calcium during that phase? So the phase 3 here, we're repolarizing the cell. We're also basically cleaning up, we're calling on all the calcium ions to get back to where they came from, which is the SR. So there's three ways of getting rid of calcium from the cell. One is via this ATPase pump. So a pump it's for active transport with the breakdown of ATP for the energy to pump this calcium against their gradient, because now they're accumulating in the SR and they're stuffed in there and they're very concentrated and we keep adding them in there. So we need a pump to push those guys back in. The calcium also gets back in using a secondary active transport using the gradient of hydrogen ions or the gradient of sodium ions. So those gradients are basically providing the energy for the calcium to be kind of shoved in this SR. And also another way of getting rid of calcium is sending it out into the extracellular fluid. And we use the same kind of secondary active transport with using the energy from the flow of sodium ions or the flow of hydrogen ions down their concentration gradient, and that energy kind of pushes calcium ions out. So I wanted to mention that calcium is being taken care of like that. Okay, so you have so this plateau, the idea of this plateau is to make sure that we prolong this refractory period where we are not allowing the cell to repolarize, or redepolarize, so depolarize again. We don't want it to depolarize again too fast. We want to make sure they contract and then they relax before they contract again. And that's why we have this long refractory period. That way, first, we avoid tetanus where the cardiac cells will just be, recontracting, and then they're just stay contracted if it's too fast in a row. And also the cardiac muscle cell will be less fatigued if it contracts, relaxes, contracts, relaxes, instead of just firing too often. So, which means contracting too often. So I'll provide you with all of the, I'll upload these diagrams for you, and then you can do as whatever you want with them. You look at the ones without the filling and then use them as your template to kind of redraw the story for yourself, or use the last one as your one final diagram to just study from. That's it. However way you want. Okay. Okay, so I just wanted to show that to you. Okay, so that's what I wanted to talk about. I wanted to talk about all these channels of ions going in and out, which one's doing what, and really how it corresponds to the action potentials. That's really the purpose of that drawing, is to show you this here, is to show you at each phase of this action potential, which channels are open, which channels are closed. And I'll be asking you about that. So make sure you know these phases. So I forgot to mention the phase numbers for the nodal cells. We start nodal cells, we start at 0, phase 0. And then this is actually phase 3. This is what corresponds to phase 3. There's no phase 1 and 2. And then we go back into phase 4, which was so that was phase 4. There we go. And it continues on about up and down, which I drew it a couple slides. Yeah, this is the slide where I drew it before, but then I don't know why it disappeared. You see how things are disappearing? That is so weird. Why is it disappearing like that? This was phase 3 and this was phase 4. There's no phase 1 and 2 nodal cells. All right. Who knows why? You see what I mean? I'm just scrolling up and down. This is not good. And that's okay. All right. So I think I jumped the gun earlier when I talked about the nitty-gritty of the conduction of the heart, because it pops back up over here. So we really don't need to get back into this. But this is the diagram that goes with the drawing I had drawn over here with the SA node, Bachmann's bundles, the internodal pathway, and the purpose is to depolarize the atria before we actually depolarize the AV nodal cells, and then that will propagate after a delay. It will propagate to the AV bundle branches, and the Purkinje fibers. All right, so we have refractory periods. I talked about that. This is really very important, so the muscle cell does not recontract too fast, right? To prevent tetanic contractions, which I already said that. Okay, a few mentions here about the differences between skeletal and cardiac muscle cells. Cardiac, the action potential is conducted from cell to cell. This is very special. That's what the gap junctions are for. In skeletal muscle cells, the action potential is conducted along the length of a single fiber. So you need a neuromuscular junction. You need a neuron to stimulate that one fiber, and that's it. They don't talk to each other like that, like cardiac cells do. The rate of the action potential propagation is slow because of the gap junctions and the small diameter of fibers. I talked about that. In skeletal muscles, it's a little bit faster. And in cardiac muscle cells, we have calcium-induced calcium release. I talked about that earlier. Movement of calcium through the plasma membrane and T tubules into the sarcoplasm stimulates the release of calcium from the sarcoplasmic reticulum. We didn't have that for skeletal muscle cells. So the movement of calcium through the plasma membrane, that is very, very, very important for cardiac cells to continue their job. We cannot count on just the calcium from the SR. This is something very important. Like if you put a heart in a bath of an aqueous solution that is void of calcium, it will eventually stop beating. So the calcium from the SR is not enough. The calcium, the extracellular calcium is super important. Okay, so we talked about atrial depolarization, which is atrial systole is happening at the time because a depolarization leads to contraction of the muscle, and contraction is called systole for these chambers. So atrial systole for an EKG. We're going to see a specific set of depolarization, repolarization, and then we're going to see abnormal EKGs. So let's start here instead of the previous slide. I should switch those around. So in EKG, and you'll probably learn more aspects of the EKG with your other professors. The EKG or ECG, I think the K is from a German, the original German word for for the cardiogram, right? Electrocardiogram, ECG. Electrocardiogram, is the recording of the electrical event in the myocardium, and we do that by placing leads on the surface of the body. And there's a great video on osmosis that explains the basics of the EKG and how these leads, each lead is basically capturing the depolarization of the cells as they're happening. And each lead is a very specific spot on the body. So it's capturing that depolarization at an angle of where the heart is and where the lead is. And the combination of all of these captures creates this very specific graph that's the EKG. So we have a P wave, we have a QRS complex, and then we have another T wave. And we're going to talk about depolarization and repolarization. The P, Q, R, S, and T, this is a very specific look to the EKG that results from the combination of all of the action potentials that are going on at the time where we're measuring this EKG. So for example, this graph you see, it's not one action potential like what we drew earlier. We drew the action potential of the nodal cell, and then we drew the action potential with the plateau of a cardiac cell. This is not the action potential of one cell. This is the action potential of all the cells combined and viewed via these different leads. So that's the tricky part, is that each lead is recording something, and And when you see the EKG like that, it's basically a combination of action potentials. So here you see a bump right here, which we call the P wave. And this bump is a depolarization and only a depolarization. So when the atria depolarizes, when they go into systole, we see on the graph a bump. So not only is it going up, but it's also going down like this. And you might ask, why is it going back down? Depolarization is when it goes up on those graphs, the action potential graphs. And then when it goes down, it's because we're repolarizing. True. That's true if we're looking at the action potential of one cell. Here we're looking at a combination of action potentials, and that first bump happens to be the depolarization of the atria. So that is atrial systole right here. So when you see a P wave, you know the atria have contracted. And then there is a delay here before you have this QRS complex. QRS complex is the ventricular depolarization right here. That's ventricular depolarization, which sets the onset of ventricular contraction. So we're going to have ventricular systole right there. And here it was atrial systole. So QRS complex, it's the ventricles that are squeezing, contracting, and they're going into systole like that. Then we have the T wave, and the T wave is ventricular repolarization. So they're relaxing, which means it's ventricular diastole. The T wave, so it's also a bump, but it's diastole and it's not systole. See, so there is no correlation, there's no oh, if we're going up it means something, and if we're going down it means something. That we can't look at it anymore like that, like we looked at individual action potentials. This is a combination of all these different action potentials, and the different bumps reflect specific events. So the T wave is specifically just ventricular diastole. Well, this is the event that's happening. So the bump itself is ventricular repolarization. Okay. So where's atrial repolarization? We talked about atrial systole, ventricular systole, ventricular diastole. So repolarization, T wave is the repolarization of that. Where is atrial repolarization happening? While ventricular systole is happening. So whatever bump we would be able to see, we would see for atrial repolarization, right? This was depolarization right here. So for atrial repolarization, whatever bump that would be is actually buried in here. It's buried in here. You can't see it because it's not as significant as the ventricular depolarization, which is what the QRS complex is. So the atrial repolarization is buried in the QRS complex. The QRS complex is mainly ventricular depolarization. All right. So the PQ delay or interval, it's that delay where we're allowing the atria to contract and finish contracting, so the ventricles are full to the maximum. At the end of that, the ventricles will start to contract. So at the end of the PQ interval, which is the beginning of the QRS complex, when the ventricles are contracting. So they contract over here, and then they finally start relaxing at the T wave. Okay. So on an electrocardiogram, you see that. You see a P wave, QRS complex, T wave. And then another heartbeat. P wave, QRS complex, T wave. atria contracts, QRS complex, ventricle contract, and then relax. Atria contract, and then ventricles contract, and then they relax. So if you look at step by step, and I included all these slides in case you really want, you're very visual, and you want to see what's happening at every phase of the ECG. And if it's just too simplified now for you, and you're like really good with this, you can skip all these slides. So atria begin depolarizing in the P wave. Now they're depolarized, they're all contracted. It's complete. The depolarization is complete, the P wave is complete. And then the signal is propagated to the AV node, down to the bundle, Purkinje fibers, blah, blah, blah. That's the QR, that's the beginning of ventricular contraction. So now the ventricles are depolarizing. Ventricular depolarization begins, and it starts at the apex, and it works like this. Remember I told you that. So the QRS complex is over here. So QR is the beginning of it, and RS is the end of it. Ventricular depolarization is complete. So that's when the blood is gone now, and they have to kind of relax, open back up, kind of like, let go. So repolarization takes place. It's the T wave. Begins at the apex and progresses superiorly as well. And then finally, we're done. Ventricular repolarization is complete. The heart is ready for the next cycle. So maybe that's when we have, passive filling of the ventricles until the atria starts squeezing, and we have a new cycle, a new P wave. Okay, so I included a few EKGs and what they look like, and I want you to think about a little bit about all of that. This is a normal one, and this one has no SA node activity. See, there's no P wave. Because SA node sends the signal to atria. Remember, the depolarization is propagated along the atrial wall, and all of the myocytes in the atria contract, and Bachmann's fibers are sending the signal to the left side, so the left side contracts as well, and it's happening at the same time. So if we don't have an SA node, we don't have a P wave. P wave is missing. Okay, so I kind of gave you a little bit the answer here. No SA node activity. What does that mean? That means that there is no P wave. You cannot contract the atria. Is it bad? Is it kind of okay? We can still survive that. Think about that. Oh, this is, we have P waves here. Where's the rest? Oh my goodness. P wave, another P wave. Okay, now we have a full one. But here we're missing. That's called the heart block. What are we missing here? What does that mean? Try to think about that. This is what a premature ventricular contraction would look like. Basically, the QRS complex would be kind of really off. And something is going on. It's called ventricular fibrillation. there's something going on, but is it enough? Is it enough? What is this? So there's a really great video about ventricular fibrillation, and when you look at it, you really realize, oh my gosh, it could be wiggly all you want. If it's not contracting, it's not pumping blood, and that's it's deadly. So I included here as a last slide some common arrhythmias, even though really this chapter is more about normal EKGs, but I thought this would be interesting for you to have. And I know this will be mentioned in other places. Yes, so I'm going to stop here. And basically, so in conclusion, I'm going to stop sharing here. So in conclusion, I covered the flow of the blood through the heart. I covered the cardiac cycle and conduction through the heart via the nodal cells, talked about the SA node, the AV node, and how this propagates down to the apex of the heart. And I really kind of dove into the physiology of these pacemaker cells' action potential, and what does that mean in relationship to the channels opening, channels closing, and I did the same for the cardiac cells' action potential, which channels are opening, which channels are closing, the significance of the plateau, etc. So I'm going to stop here. Thank you.